Welcome to Going Antiviral, the podcast of the IAS-USA, a professional continuing medical education organization focused on HIV and other viral diseases. I'm Dr. Michael Saag, professor of medicine and infectious diseases at the University of Alabama at Birmingham and volunteer member of the IAS-USA Board of Directors. Welcome to the Going Antiviral podcast. Today, December 14th, 2023, we're speaking to Dr. Anthony Fauci, former director of the National Institute of Allergy and Infectious Diseases of the National Institutes of Health, and currently distinguished professor at the Georgetown University School of Medicine. Dr. Fauci grew up in Brooklyn, New York, worked in his father's pharmacy, and played basketball and baseball at Regis High School. In 1958, he enrolled in the College of the Holy Cross, graduating in 1962, and then attended Cornell University's Medical College, graduating first in his class with a Doctor of Medicine degree in 1966. He completed his internship and residency in internal medicine at New York Hospital, the Cornell Medical Center, and after residency in 1968, Dr. Fauci joined the National Institutes of Health as a clinical associate in the National Institute of Allergy and Infectious Diseases, or the NIAID, Laboratory of Clinical Investigation. He led the LCI Clinical Physiology section in 1974, and in 1980 he was appointed chief of the NIAID's Laboratory of Immunoregulation. His work in the 70s, including groundbreaking discoveries in the treatment of many rheumatologic diseases, including polyarteritis nodosa, Wegener's granulomatosis, and lymphangiectatic granulomatosis. His work formed the foundation of treatment of these diseases over the last half a century. In 1984, he became director of the NIAID and retired from this role in late 2022. Tony, welcome to the podcast.
Thank you, Mike. Good to be with you.
So let's just start with the AIDS story. When did you first hear about AIDS, even though it wasn't called that initially?
You know, Mike, I remember it crystal clear, like it was yesterday, and that is I first heard about it without realizing what we were dealing with literally in June of 1981 when the first MMWR landed on my desk in my office in the clinical center of the NIH in Building 10. I looked at it. There were 5 otherwise previously healthy young gay men, curiously all gay, who presented with pneumocystis pneumonia. I thought it was a fluke, to be honest with you, Mike. I didn't think it was going to turn out to be anything, and I thought it would be a one-off. Perhaps they took some drugs that was suppressing their immune system that allowed them to get opportunistic infections. But then a month later, in July of 1981, my whole professional world turned around because I saw the first MMWR, now of 26, curiously all young, otherwise healthy gay men, not only from LA, but now from San Francisco and New York, who presented not only with pneumocystis, but with Kaposi sarcoma and other opportunistic diseases. And it was at that point, very soon after reading that July 1981 MMWR, that I literally changed the direction of my career and began studying this mysterious disease without a name and without an etiologic agent.
And when did the first patient or two arrive at the clinical center at NIH?
Well, when I decided that we were going to essentially change the direction of my research, which was, as you mentioned correctly, Mike, was essentially developing therapies for autoinflammatory vasculitides, I needed some help because I knew we were going to bring in a lot of sick patients. Cliff Lane had just finished his fellowship in my lab, and I asked him if he was interested in joining me in this new endeavor, which he was enthusiastic about. We needed an intensive care specialist, so I recruited Henry Mazur from the New York Hospital because Henry was a medical student when I was chief resident there. And then he was one of the first people who actually described the first AIDS patients in a New England Journal of Medicine paper back in the early 80s from New York. So we decided to bring in patients. And a few months later, the first inpatient that we brought in, I remember very clearly, was a person who had an identical twin. And what Cliff and I and Henry decided we would do, quite naively in retrospect, was to see if we could take the bone marrow from his identical twin and transplant him or give him lymphocyte transfusions, we might be able to essentially cure this mysterious disease, not realizing that the existing virus would very quickly knock out the transplanted bone marrow and the lymphocyte infusions. But that was one of the first patients that we admitted.
Well, I didn't know about that and how prescient in a way, because the bone marrow transplants done under the cover of antiretroviral therapy have led to the few cures that we've seen. That's interesting. In 1984, you become director of the NIAID, and you're in the position now of moving resources toward the study of HIV, its treatment. Tell us about those mid-1980 times and what your thoughts were as you put all this together.
Yeah, Mike, the reason I accepted the job at a very young age as the director of NIAID was because, and I didn't seek or aspire to be a science administrator. I was, like you, very, very much involved and happy with seeing patients, multiple patients on a daily basis, and doing research in the lab. But I just felt strongly that the entire field of infectious diseases was getting short-shifted in regard to the supply of resources. In fact, NIAID at the time was sixth out of about 15 institutes, 12 institutes at the time, with resources, even though, infectious diseases globally was the second leading cause of death worldwide. And in some even developed countries, it was one of the leading causes of death, particularly among young people. And I felt strongly that we weren't putting enough effort into HIV/AIDS. So I accepted the job under the condition that I would maintain my lab and maintain my contact with patients, which thankfully the director of NIH at the time, Jim Weingarten, agreed that he would allow me to do that. And that's of note that was the first time any director who took the job as director of an NIH institute actually continued to see patients and had a lab of their own. So it was the exception, which is now the rule, that most institute directors at least have their own labs. Not very many see patients, but they have their own lab. So that's the way things were in the 80s. But, one of the things I described, Mike, is that taking care of patients from 1981, and I know you had the same experience, through the late 80s into the early 90s, were really in many respects the dark years of our professional career, because we were trained to be healers and we weren't healing anybody. We were putting Band-Aids on hemorrhages. And I remember, the whack-a-mole we used to play with OIs. You have somebody has pneumocystis, you treat it, he gets better, they get toxo, you treat it, they get better, and then they get CMV or something else and they die. And that was very disturbing, very painful. And we all felt that. And then when we started to get drugs, then things started to turn around.
So the first drug was AZT, and I think it originally came out of the NCI with Sam Broder, Bob Yarchuin, and others. But you all worked closely together, is that right? And
yeah.
tell me about that.
So what happened at the NIH, that we had a couple of groups working on this new disease. I was doing it with Cliff and Henry from a pathogenesis standpoint. Bob Gallo, as a retrovirologist, was obviously interested. Could this possibly be a retrovirus? Because as you know, in 1979, Bob had identified HTLV1 as a cause, the etiologic agents of human T-cell lymphoma leukemia. But what Sam Broder, Mitch Mitsuyu, and Bob Yarchuin were doing, they developed a simple reproducible assay to let the virus grow in culture. And they began screening already existing drugs. This was not a targeted drug development program. It was screening existing drugs because we needed something right away. They partnered with Burroughs Wellcome, who had a whole list of drugs that interestingly were failed drugs for cancer. So AZT was developed as an anti-cancer drug and didn't work on any cancers. But Bob and Sam found out very quickly that, to their great surprise, suppressed the replication of HIV in vitro. That led, as you know, to the first NIH-sponsored trial together with Burroughs Wellcome of the first and only placebo-controlled trial of a drug of people who had AIDS. And you remember the criteria to get into the trial is you had to have had a prior bout of pneumocystis pneumonia, recovered from it, and then you either got placebo or you got AZT, and the results were striking. For the people who got AZT, there was 1 death. For the people who got placebo, there were 19 deaths. And that was leading to something that I find very gratifying. It was the fastest approval of a product by the FDA in its history. It essentially approved it in a matter of months.
And that's something that is remarkable, but yet, despite that speed, despite that effort, the patients, especially in the activist community, didn't feel like much at all was being done. And they were looking around seeing friends die, and they came to NIH, you, others, just to put more pressure on the speed of development. Let's talk briefly about the role of activists and how they spurred us on in a way.
Well, the activist role was highly productive, beneficial, and all in all, a very positive phenomenon. I must say parenthetically, Mike, very, very much contrary to the anti-science activism that we have today. In fact, the AIDS activists were pro-science activists. They wanted 2 things. They wanted more resources to be put into the AIDS effort, but they wanted to be part of the discussion of what the most appropriate clinical trial design would be. They wanted flexibility in the regulatory approach to the approval of drugs. They wanted to be listened to. They wanted a seat at the table. And I must say, we in the scientific community early on were not listening to them because we stood by the old adage, which has been proven to be incorrect, that we scientists know better than anybody what's good for the patient, as opposed to the patient playing a major role in the discussion. And it was only when I started to listen to their iconoclastic, disruptive, theatrical behavior, because we weren't listening to them, that when you put aside all of that and listen to exactly what they were saying, they made perfect sense. And I asked myself, if I were in their shoes, I would be doing exactly what they were doing. And that's when it all started, when we began to sit down in the conference rooms with each other and talk about how our common goal was to appropriately address AIDS. And over the years, the activists became important parts of our advisory councils, our community activist programs, to the point that they were now a seamless part of the effort. It started off confrontationally, but it wound up really, really productively.
Yeah, and you became close friends with a lot of them, most notably Larry Kramer, who was an incredible, energetic, and forceful personality that made a big difference to a lot of people. But I think mostly you, and you're in the position now where you can generate resources within the NIAID, go to Congress and get resources to fight AIDS. You were very successful at that. And you created the ACTG, the Trials Unit for Drugs, HPTN for prevention, the HVTN for vaccine. And you also worked with the FDA a lot in doing what's called the accelerated track. And I'm mentioning all this because it all lays the foundation for what happened later with COVID that we're going to get to in a second.
Yeah. Well, there are a couple of things that I might mention for what you said, all of which were quite correct. Is that when I started the division of AIDS, there was a lot of pushback, interestingly, from other of the older members of the infectious disease community who felt somewhat threatened that if we had a separate division of AIDS, we would take away from the resources that were needed for other infectious diseases. And I insisted, and I was correct, it turned out, that we could get more resources and not take away anything from malaria, tuberculosis, or influenza or diarrheal diseases, which was the case. And as it turned out, the AIDS budget turned out to be 10% of the entire NIH budget, and no other infectious diseases actually suffered at the same time as we attracted a large number of very talented investigators to get involved in the AIDS field.
And through that, the revolution happened, if you will, in the mid-1990s, some of which led by the development of viral load assays, that we didn't have to do body counts anymore like was done in the first Burroughs Wellcome study. Rather, we could tell in weeks whether a regimen or combination regimen could drop viral load sufficiently and sustain it. And by the time we get to today's world, most people can readily gain access to the medications. When they take them, they work spectacularly and have few side effects. And AIDS has been converted from a death sentence to a chronic manageable disease, thanks to the efforts of all involved. But I would also give a tip of the hat to the activists who really pushed all of us to work harder and faster. Let's fast forward to January 2020. You have been working over the years on a number of outbreaks, including influenza of different sorts, the original SARS-CoV-1, Ebola, and you find out about this new entity called SARS-CoV-2. We spoke with Rick Kalp, and we can go to that podcast to get details about the original setup that you and others created through the National Vaccine Research Center. But how did that affect you personally when you first heard about COVID and what you were thinking as this started to evolve?
Well, in the very first weeks of January, I mean, the story is an interesting story, I can tell you, because we had been very much discussing the possibility of making a vaccine for mares. Barney Graham had made a major discovery in showing that if you stabilize the pre-fusion F protein of RSV, you could develop a very highly immunogenic, effective RSV vaccine, which, quite frankly, was the basis of the RSV vaccine that was just recently approved. But when we used that technology with mares,
MERS being the Middle Eastern respiratory
Middle East respiratory syndrome, which was also, a coronavirus.
Mm-hmm.
So what happened that literally in a few days after the 1st of January, when we found out about it, Barney Graham came to me and said, Tony, if we get the sequence of this virus, we could start a vaccine work within days, because they had already been working with Moderna on the mRNA technology. And sure enough, when on January the 10th, the sequence of the virus was published in a public database, I said to Barney, let's go and do it. Barney, a typical great scientist that he is, said, I can do it, but it's going to take money. And I said, don't worry about the money. I'll worry about the money. Let's do it. And Barney and his team, together with Moderna, as well as the Pfizer people, embarked on something that was historic. They went from getting the sequence on January 10th to starting the actual vaccine development with Moderna 5 or 6 days later, to a phase 1 trial 65 days later, to a phase 2 trial 100 and some odd days later, to a phase 3 trial 190 days later, to 11 months later, having completion of a clinical trial involving tens of thousands of people that showed that the virus was not only safe, but more than 90% effective going into the arms of individuals. So that first couple of weeks was the initiation of a historic vaccine trial, which is what the NIH does. I mean, we're not the CDC. We're not a global health organization. We're a scientific organization. And I think what we did with the vaccine was something that really is a model for future approaches towards outbreaks.
Absolutely. And it was termed Operation Warp Speed. And a lot of it was built on the foundation that you and many people around the country and the world had developed with the ACTG and the other trials networks. And you plugged these studies into that kind of model, also working with the FDA. So
Right.
Maybe you could say how the back channels went as you threw this together with a lot of other people.
Well, first, let's talk about the network that you referred to. As you very correctly just mentioned, that for literally decades, we had put together networks of ACTG, HVTN, HPTN. We had a number of investigators, mostly in the United States, but throughout the world, who were absolutely well trained and experienced in doing clinical trials. As soon as we needed a clinical trial network to test the COVID vaccines, instead of reinventing the wheel, we leveraged the already existing clinical trial network, and we had the interesting but gratifying situation of a whole bunch of AIDS researchers doing clinical trials on COVID vaccines. And that's exactly what happened. We again worked with the FDA. We were on conference calls literally on a weekly basis with the companies that were doing the trial, the investigators that were doing it, the FDA, the CDC, and the NIH to make sure that those clinical trials were done in a way that there was equitable access, so we had good minority representation, that the trials were done correctly. It was really an amazing and very gratifying example of collaboration across government institutions, across the academic institutions, and together with the international society.
And the pharmaceutical companies.
The pharmaceutical companies were total partnership, partnership.
And so you mentioned garnering the resources to fund the trial, but in parallel, if you have a successful trial and a vaccine with 90+% efficacy comes along, that doesn't mean you'll have product at the end of the study. So in parallel to this, you and others are working on funding development of plants to manufacture a product that you didn't even have a clue yet would work.
Well, that's what's called high risk. No, so what happened, Mike, is that was the strategy that led to the success of Operation Warp Speed to get a vaccine into the arms of people within 11 months of the identification of the genomic sequence of the virus. So here's what happened. As you well know, you were involved in many clinical trials. You will know you do a phase 1 trial. If it works, you go ahead to a phase 2. If that works, you get to a phase 3. If you have a good product, then you start manufacturing it because you know it works. There's no risk to the company. They have a product that they've proven in a phase 3 trial works. That's not what happened with COVID. They started manufacturing the product as we were doing the phase 1 trial. Amazing. An incredible financial risk. Now, the only good thing about that is the financial risk was on the part of the federal government. because it was the federal government that put the money in, not the pharmaceutical company. But the risk was great. So it was a typical example of a high risk, high reward activity.
And we talk about these all the time and try to find them in science, but this one just unfolded before all of our very eyes. And that was quite remarkable, such that in late November, early December of 2020, the results are released. And within 3 weeks or so, I got my first shot. So that just blew me away that we could have an understanding of this, demand for it. Others maybe had to wait 6 months if they were part of the general public. But yet, within 6 months, the majority of people who wanted a vaccine got one. We only have a few minutes left, unfortunately, but I do want to circle back to some comments we talked about earlier regarding the role of activists. And you mentioned that the HIV activists were not only helpful, they were smart, they were well-informed scientifically, and they spurred the development of all kinds of things, including this expanded access and parallel track development, which got drugs that were successful available to people before they received FDA approval. Yet in COVID, it feels like we have the exact opposite. And I just want for you to just reflect for just a couple minutes on what this was like for you, organizing all this response that you just described so nicely, when you start getting attacked personally and otherwise by these so-called activists of the COVID scene.
Mike, it was diametrically opposite to the experience with HIV, which was activism for a good cause to make things better. What we were facing with was an anti-science, anti-vaccine, conspiracy theory-driven amount of activity. Attacks on scientists, not only myself, but a lot of public health officials that were trying to do the best thing for the public, talking about the need for masking, the need for the development, and the need for the acceptance of vaccines. Not only were they responded to negatively, but there were threats and intimidation and attacks on many, many scientists, including me. I'm a very visible person, so I was the one that got a lot of that. But many of my colleagues also, who were talking about proper public health principles, were getting attacked. So it's a totally different kind of activism, one that's destructive and has a negative impact as opposed to the positive impact that we saw during the years of HIV.
And that what I called it was not literally, fortunately, but at least figuratively, the assassination of the trusted voice, where you or others are trying to get messaging to the public, just like a doctor tries to get a message to their patient. In the case of a public health official, they're trying to communicate to the population who is collectively their patient. And who would ever think to question a doctor's good faith in interacting with their patient and understanding as a patient, if the data changes, the opinion changes, and we've got to roll with that. There was no tolerance of that among a lot of people during the COVID vaccine.
It's called flip-flopping. And, and as we know, we say it very often, science is self-correcting. Science is a process. And the process is to accumulate evidence and data and to make your recommendations based on the best possible data that you have. And when you're dealing with a moving target, and Lord knows if there ever was a moving target, is SARS-CoV-2 with 6 variants and now multiple Omicron subvariants. It's a moving target. And to say that you have to have static recommendations, and if you change anything, you're flip-flopping, is just complete misunderstanding of what the scientific process is.
And again, it's just like a doctor who is seeing somebody for fever. They do an initial evaluation, give you a list of possibilities, but that changes as more data come back. And that's exactly what you guys are doing. We got about a minute. Maybe you could just reflect on what do you think we can do to combat this negativism and this antagonism that we feel from some sectors in not just the United States, but around the world?
Well, Mike, I don't know, a definitive great answer to it, but I strongly suspect that as you have so many people who actively and energetically spread false information, that we in the scientific community have to put a lot more energy into spreading correct information. We can't prevent people because of freedom of speech of saying what they want to say, but we've got to put as much energy in being out there talking about what the facts are, what the evidence are, and what the data are, because if we don't, the people who are spreading untruths are going to outflank us. I mean, that's what's going to happen. And we can't let that happen.
So we will all dedicate ourselves to doing that in the coming months and years ahead. I don't think it's going away, but hopefully we can get a foothold and turn this around. Tony, this has been a wonderful conversation and involving your truly remarkable career so far. I know you're still at it, even though you're no longer at NIAID. I wish you the best. I congratulate you on all that you've accomplished, but mostly I thank you for the incredible spirit you've had and the leadership you provided, especially to the scientists in the community and for me personally. So thanks so much.
Thank you very much, Mike. It's a great pleasure to be with you, and it's always good interacting with you. Thank you.
Thank you for listening to this episode of Going Antiviral. This podcast is for informational and educational purposes only and is not intended to serve as medical advice. Opinions expressed in this podcast do not necessarily reflect the opinions of the IAS-USA or its affiliates. To listen to more episodes of Going Antiviral, be sure to visit our website at IASUSA.org.